Okay, thank you, Gonzalo, for this kind introduction. So, as already said, I'm a PhD student at the Department for Chemical Sciences at Sapienza University of Rome. I just want to say that we are a multidisciplinary group that is working to synergize structural and cellular biochemistry with computational models. In this webinar in particular, I would like to delve into the recent advancements in our field through the point of view of structural bioinformatics tools that are available in our field, of course. Here, just a brief great introduction. Can I in some way remove that? Oh, sorry. Okay. Just a brief introduction that I would like to delve into this webinar with a division that I like to make between a pre-AlphaFold and a post-AlphaFold area. But I would like to use AlphaFold just as a chronological watershed between these two areas, given the number of things that have changed since that release. But other than the great achievement itself that I'm not going to solve now because we all know, and it is not the focus on this point, I would like to focus on two other features that also made AlphaFold what it is today. So first of all, it's the code release that puts AlphaFold in a collaborative mindset because it has boosted also the, let's say, integration of AlphaFold into different protocols and also the development of related tools. But not forget also the development of the AlphaFold database that promoted the accessibility to structural data. In a certain way, this allowed also everyone to obtain a computational model of the protein of interest with a minimal effort. So this broadened the accessibility to different type of research, but we started to reason about how this broader accessibility can influence different type of researchers in different ways. With this, I mean that on one hand, it can be useful to broaden the accessibility to and to gain structural insights also at in different levels of research, different type of research that was not thoughtable before. But it is still, we think that it is still important to leverage on those aspects that can guarantee for a proper interpretation of the model itself from, let's say, a biochemical perspective. I feel to say that because with the widespread adoption of and success of AI and computer science techniques in biology, the field continues to create a multidisciplinary environment. So we must consider that there are going to be people that are going to transition to our field as also new students of the recent years who are only going to be witnesses of this post-AlphaFold area. But I think that there are those people that have worked on this also before, that this type of insight that they are going to, that they are able to reason on still must be useful insight also in this post-AlphaFold era. And so even if there is a huge, let's say, speech that can be done on this part, from our side, we started to reason from the perspective of the development of tools for assisting structural bioinformatics by reasoning on the features that could help in conveying the biochemical principles which are underlying the prediction of the structure of a protein. So I can understand that there is little room for improvement in some cases in trying to increase the modularity and the accessibility of environments that implements black box models. So those models are only from an input give an output like AlphaFold. But I still think that it is important to harness software developments that facilitates a unified application of structural bioinformatics protocols, and it's in a certain way also fostering a user-driven approach to this kind of questions. So first of all, I have started to analyze the tools that have been developed in our field from the bio tools annotations. We can say that there is a sort of pattern in the development of course, web applications and command line tools. We know that web applications are are a lot popular because they are easy to access, but we may also say that they are rely on servers, and sometimes you don't have so much control over them. But the other way, we still have command line tools, but we know their importance, and of course, but I would like to focus the attention a bit more on the desktop applications that still seems to be rare in this field, maybe because there is a certain, let's say, some hampers in in their development, but we can come to this back later. In general, this was just a brief introduction on the type of, let's say, schema that we have in the development of structural bioinformatics tools, but I don't think that we can, and we neither should tell which is the best way to deploy a structural bioinformatics tool, but we could reason on the features that can make an application open to a user-driven approach and also to be training friendly. So here I have, for instance, set some ingredients of this recipe, we can say. First of all, I think that we need to avoid the development of blind software. So those applications that takes some inputs and produce outputs without requiring the user to understand the underlying process. While this approach can promote in a certain way efficiency and caters to user of different levels of expertise, in a certain way can risk to discover a deep understanding of the methodological principles underlying these processes. On the other hand, I think that it is really important to promote tools that enable the user to have a control on the input itself, and also on the processing of the input itself. And another important thing is that since we are in a field that it is primarily rich of a diverse set of tools, also the integration in a unified environment could be of utmost importance. With this introduction, I want to introduce you with a focus of our lab, which is the development of PyMOL plugins. One of these plugins that is much more used in the also our training platforms. But sorry, I don't know if this is hiding something.
No, you only see it.
Okay, perfect.
For us, we don't see the plugin there.
Okay. Um, so PyMOL-3 is known as a complete suite for structural bioinformatics in PyMOL. It is a desktop application. And in here, I want to add some other two ingredients to this recipe, because even if these three can also be broadened to different type of softwares, but there are something which are peculiar also for our field. In this case, I think that it is really important to, when possible, to implement a way to promote the editing of multiple sequence alignment. This can, from a side, help researcher to faster this process. On the other way around, we saw that it is really important for new students to understand what also these simple steps that sometimes are in a certain way overshadowed because they are just done by server on some cases. On the other way, it is also important, and for this way, distinctly we exploit PyMOL because we like the possibility to directly exploit a very common graphical user interface for the graphical visualization of proteins. And in this way, since PyMOL is implemented directly in PyMOL, we can also have to the mapping of some analysis on the protein itself. So in order to show some other features of PyMOL, I really like to use this kind of example. So this is a very huge protein that comes from Pseudomonas aeruginosa. From a biological perspective, it is important because some structure inside of this protein can give important insight for the treatment of cystic fibrosis, but I don't want to go into the details of the biological principles. What I like to use for this example is that it is a multidimeric, multimeric protein, a transmembrane protein. So in some ways, it can be difficult also to just rely on some predictions, for instance, the predictions of AlphaFold. So we can understand how we can approach it in this unified environment. So I'll start again this video. So let's imagine that we just have the sequence of this protein and we want to understand whether AlphaFold could help us in gaining some structure inside of this protein. So directly in PyMod, we can get the protein, the model itself. From an expert view, we can already see that there is something that is not very pretty accurate of this model, mainly for the interdomain positioning, and we can continue to also confirm this speculation by plotting the predicted aligned error plot. This, of course, confirmed that in this case, it is not possible to use the AlphaFold model directly. So how can we approach this situation exploiting the environment of structural bioinformatics tools? For instance, we can approach this with a domain-driven perspective. So we can approach it by scanning with the HMM scan for domains that are notated in PFAM. So we can say that with this analysis, PyMod features this graphical user interface to have to show the domains that have been found, and you can select those to which we are interested. Once you click, so you select the domain itself, and once you click on submit, the sequence is color mapped. I started this because at this point I would like to show how we can, let's say, of course, I don't have the time to show the process to the building of this entire model, but I can show how someone could approach to this, for instance, for the modeling of just two domains, and then to broaden this process also to the other domains. So still starting from the domain analysis that we have done, we can exploit directly PyMod in order to just retrieve the domain of interest, in this case, these two domain paths at ggdef. We can exploit the editorial sequence of PyMod in order to cut the sequence that we do not need now, and just keep to work with the other part of the sequence. So I'm just skipping a bit. So we see that now we have the sequence that has been cut. I must say that since this sequence has been edited, we still will need to do again the PFAM search, so to look for the domains. And of course, now we only have the domains of interest, so path and ggdef domains. At this point, this part of the model can be let's say, can be approached by splitting into domains. So working at the beginning by splitting and looking for some structural templates of a single domain and the other domain. So we can split the sequence. We'll see that we can also choose for the overhang of the sequence. This is important because this allows us to have an overlap between the two domains. That will be important in the next steps. So now we see that we have two new objects in PyMol showing the sequence and the domains. We start by looking for some structural templates by running a local blast on PDB. I just want to repeat that I'm not going into the details of the reason to choose some templates with respect to the other, but what I want to focus the attention on now is that it is still important also to do not put these, let's say, these steps of the structural templates of choosing the structural templates as an automated manner, but it's important still to choose and to have a platform that allows you to choose it with a graphical user interface, not just by, for instance, a percent of sequence identity without the user to put its hands on this process. So at this point, we choose the structural templates of interest, and we can do the same things with the other sequence that is present in our environment. So we still run a blast search, and we select another structural template for this protein, for this other subdomain. I will skip a bit. So now we have in PyMOL these two clusters, which are the two domains aligned to the structural templates that have been chosen. Since we still have the original sequence, we can now fuse these two domain alignments into a single one. Also because we have the overlap of these two sequences, and also the structural templates are going to overlap on that part. This is done, I don't know if it was too fast, but this is just by clicking on the starting sequence and click on fuse domain alignments. At this point, we can, first of all, retrieve the structure templates that have been chosen. This is the I think that now I will skip a bit because we are just cleaning the environment because we do not need anymore the splitted sequence of the two domains because we are going to rely on the entire one. So here I'm just deleting the sequence. I'm going to skip this part a bit. At this point, we can retrieve the structural templates, just the it part, so just the part that has been found to be the template for that particular domain. And we, by clicking on the original sequence, we can now run, for instance, an homology modeling by exploding modeler. In this case, I have found that the best results are given by choosing an optimization level high, and this objective function that has been developed in our lab, which adds statistical potential to the objective function of modeler. And we see that it is very precise in creating the final model. So I'll also skip a bit this part. So this is the graphical user interface to set up modeler options. Also in this case, another important thing, of course, is that PyMOL implements the modeler itself in a intuitive environment. We can continue with that. Okay, this may take some time, so I'm going to skip that. Okay, and so when the models are ready, we can exploit the dope score of this model. I'm going to skip that. And then we can exploit directly PyMOL in order to show the structure inside and show the models themselves. Here I'm cleaning a bit the workspace. So I'm removing the structural templates and we will have the three models that have been made with modeler itself. Okay, so this was just, let's say, an introduction to show how in a single environment with few clicks, we are able to make the predictions of course, exploiting homology modeling, but make the prediction of two different domains of the same protein. As I was saying, I don't have the time to continue, but of course, with the same logic, we can continue to create also the model by adding the new domains. What I wanted to say that I did an example by exploiting the structural templates that are found in the PDB, but of course we could also think about if these structural templates are not available, we could also think of using some parts, for instance, of the AlphaFold models or some other models that are present in other cases. For instance, those parts which maybe can show the highest PLDDT score, so we can have an highly accurate model of a part of the protein, and that can be exploited in combination with some of the protocols, which are, let's say, the driven methodologies in the anti-AlphaFold era, in the pro-AlphaFold era. And in combination, we can create a complete model of a protein in an integrated environment. So here I had the opportunity, so I'm still showing that we can continue with adding some other domains, so the transmembrane domain and the periplasmid domain. And I've had the opportunity to show some of the features of PyMOL, mainly the domain assignment and the domain fusing and homology modeling. But of course, there are a lot of other steps that can be done in it. And here it is a list, but it is not complete. For a comprehensive list, please refer to the PyMOL user guide. I would like to thank everyone for this opportunity. But I would like to first hand with this let's say, description of the research software engineer that I've found online. I like it very much because it conveys the message of how much it is important to have both, let's say, a background on the biological part, so on the structural development, but also to have expertise in software development in order to make this tool really user-driven, intelligible, and training-friendly for everyone. I would like to thank the main developer of this third version of PyMod, and then I pass the, let's say, the part of this research software engineering to a new master thesis that is now working on this, and that this is working also to continue to implement, because we still think that also in the post-AlphaFold era, it is still important to harness on the software development from this kind. So thank you to everyone.